Hello, next section is Unit 1.4, Cellular Differentiation. So we're moving away from all that, the kind of DNA material, and I suppose the first three topics, it's all kind of, you can kind of put all of that together, I suppose, structure of DNA and then DNA replication and then turning DNA into protein. So slightly different this time. It's called cellular differentiation, but it's essentially stem cells. So for this, explain what cellular differentiation is, what that means, and then we're going to look at meristems, explain what they are, and also stem cells. And you remember stem cells was in National 5, so you've already got a bit of background knowledge on that. So here is your going back to the very beginning. So here is a sperm fertilizing an egg cell. You've got your zygote, and then as the embryo develops, the various different cells will come into being: nerve cells, red blood cells, fat cells, etc. Okay, so you start with one zygote, and from that the cells differentiate and you get various different types. So cellular differentiation, put simply as a meaning, is the process in which a cell develops specialized functions. So you think about your original zygote is unspecialized, but then over time it becomes heart cells, skin cells, liver cells, etc. So that is cellular differentiation. So it's when the cell develops specialized functions. And the way in which this occurs is that specific genes are expressed within these different cells or switched on. So if you look at, if you compare any two of the cells in your body in different parts, whether it's in, your brain or your intestine, you have those same 46 chromosomes, you have those same genes. However, depending on where that cell is in the body, you're going to have different genes which are switched on or expressed, and the result of that is that you have cells which do different things. Okay, so cellular differentiation, you might be able to get some of this. So unspecialized cell in animals, those are your stem cells, and in plants they are called meristems. Okay, now in these cells, when they're unspecialized, all genes can be switched on, so they can become anything. Then when you've got a specialized cell, when cellular differentiation occurs, you then have various different types. So an example, neurons in animals, you've got your phloem in plants, and in these circumstances, in terms of genetics, only some of the genes can be switched on. So these cells have more limited uses, limited functions. In plants, there's meristems at various different locations. And now let's have a look at, there's lots of stem cell videos, but I've kind of picked out what I think are the sort of best ones on YouTube. So have a look at these. What are stem cells? And then how do stem cells work? And that will lead you into the next part. So we have two types of stem cell, as was mentioned in the video. So the first one is embryonic and the second one is adult or tissue. So embryonic, of course, is going to be in early embryos, and if you have them in laboratory conditions, then they can self-renew, and that basically just means that they copy themselves. In natural conditions, what they can do is they can become any type of cell. They can differentiate into any type of cell. So that's called pluripotency. So embryonic stem cells are pluripotent, can become any type of cell. So we're going back to just after conception here, the beginning of life. Now the other one, adult tissue stem cells, which we all have, they're found in the body tissues of mature organisms. So in terms of cell division, they can copy themselves, and differentiation, so they can copy themselves naturally, and what they do is they become a limited number of cell types. So rather than pluripotent, we call adult or tissue stem cells multipotent. So they replenish differentiated cells that need replacing in that tissue. So it's a kind of convoluted way of saying, if you cut your finger, then you're not going to have an open wound for the rest of your life. The blood vessels are going to heal. You're going to have new skin that grows over it. And those new skin cells are being produced from the adult stem cells that are in that area of your body. Just a wee diagram to show this. So there's your fertilized egg. At this point in time, this is a mass of stem cells in these embryonic cells, and over time these develop in a rather short space of time. You have the various genes switched on, switched off, and you have different types of cell, and they eventually make up the different systems within your body. Again, just a wee diagram to show you what kind of stage we're at here. So here we've got the fertilized egg is moved down from the fallopian tube and it's implanting into the wall of the uterus, and it's at this point in time that we were all a mass of stem cells. And you can see up here, it's not very long into the process when they start to become specialized, I'm about to show you, but this is embryonic stem cells at this stage. Okay, wee task for you. So take plenty of space and I want you to draw this. So just leave space inside so you can write a few words within each one, or a few sentences, I suppose. So press pause and do that. And what I would like you to do, based on what I was talking about, and probably those videos helped as well, I want you to copy these into the correct section. Okay, whether it's embryonic stem cells, both, or the adult stem cells. So press pause and complete that task. So this is what you should have for embryonic stem cells. They are pluripotent, which means they can differentiate into any cell type, can self-renew under laboratory conditions only, not naturally, found in early embryos, and their use is controversial. And we'll come on to this controversy in a minute. Both of them, they're both found in animals and they're unspecialized cells. And then for the adult stem cells, multipotent, capable of self-renewal, found in the body tissues of mature organisms, differentiate into a limited number of cell types, and replenish differentiated cells. And that's the first section of this, so just what cellular differentiation means and the differences between embryonic stem cells and adult stem cells. So time to go and do something else. Right, to begin with here, see if you can remember this from when you last did the section on stem cells. So press pause and see if you can fill in the blanks from memory, ideally. So embryonic, early embryos, number 1. Number 2, cell division, they can self-renew in the lab. 3, they can differentiate into any type of cell, which means they're pluripotent. Number 4, your adult stem cells, they're found in the body tissues of mature organisms. They can self-renew and their differentiation is limited. So they are what's known as multipotent, and so they differentiate into a limited number of cell types and they replenish differentiated cells needing replacing in that tissue. So remember the example I gave you was if you had cut yourself, then the new skin cells which cover the cut are from these adult stem cells. So just moving on a little bit, we're going to look at the therapeutic and research values of stem cells, so what they can be used for in therapy to help people, and also research essentially to find things out, to learn more about the world around us. And number 4, describe ethical issues surrounding stem cell use. So various different therapeutic uses a lot of this is still kind of ongoing at the moment, but anything where you need to replace cells is potentially use of stem cells. Blindness, injury to spinal cord, if you could replace the tissue there, get the nerve connection so people are not paralyzed anymore. Various different uses. Okay, but it all comes under the sort of banner of repairing damaged or diseased organs or tissues. Skin grafts is another one. One of the problems, you might be aware of this, if you've got if somebody needs, say, a kidney transplant, they always try to get a kidney from someone that's close related to them, and the reason is because the related person is going to be genetically similar. However, people who take who receive an organ transplant, they're on kind of immunosuppressant drugs for the rest of their life, because essentially the body thinks this is something foreign and it tries to get rid of it. So if you could use stem cells to grow grow your own kidney using your own DNA, then that would get round that issue of people rejecting organs and having to be on drugs for lifelong, so that their organ isn't rejected. Okay, here's another YouTube film. Have a look at this: therapeutic stem cells. And we're now going to move on to the research uses. So what they can be used for is just a way of finding out how cells grow, divide, differentiate. Cancer is a kind of obvious one, because what you've got there is really mitosis has gone wrong. The cell is dividing uncontrollably. Now, what you could do with, if you had therapy which could target the stem cells which are causing that tumour, then that would, specific targeting that could reduce the growth of a tumour. Whereas conventional cancer therapy, it just kind of kills all the cells, so you may leave some of the stem cells, which would lead to the tumour relapsing. So that's the first research use, for finding out more about how cells grow and divide, the process of mitosis and differentiation. You might recognise this man. That is Stephen Hawking, and he had motor neuron disease, or as they tend to call it in America, ALS, which is basically your motor neurons. They start to die, basically, and you lose kind of your voluntary functions. Something else you could use stem cell for is a model to see how these diseases develop. So in this study here, they've taken skin cells from a patient with motor neuron disease, and they've looked at, potentially what is the maybe the genetic issue, or what is it that's causing those cells not to function in the way that they should. So model cells to study how diseases develop. And the final one is drug testing. So essentially what tends to happen in terms of animal testing, mice and rats are very commonly used. The reason for that is they're mammals like us, so they're fairly similar, but obviously they're not humans. And if you're carrying out drug trials, you want it to be on, it's the very last stage, which is in humans. But if you could grow, say, human tissue using stem cells, then that would speed up this process. You could get drugs out in the market to help people more quickly. You wouldn't have to kill as many animals and so on. So if you imagine, what you could do is, if you could get stem cells and you were able to program them in such a way, switch on the specific genes needed to produce a heart, then you could have human hearts that you apply the drugs to, and you would test them rather than testing so much in animals. So here's an example here. So you've got a patient's cell, so it's reprogrammed to produce what's called induced pluripotent stem cells. With these cells, you can make anything, basically: cardiac cells, liver cells. You can test the cells directly. It might be possible to make them into human tissue. But yes, drug testing using stem cells is something which may come. And then finally, the controversy. So stem cell research in this country is legal. In other countries, it's not allowed. And with this, it kind of goes back to people's belief in our thoughts on where life begins. So I just wanted to show you this kind of pictures of or um, representations of a developing embryo and what kind of life looks at various stages. You know, you can have a think about what you believe yourself. So when we're talking about stem cells, we're really back here. So very early on, so the first sort of few days after sex, basically after conception. So this kind of ball of cells, the very first few days before a woman would know she was pregnant. This is what we're talking about here. So at this stage, this is 4 days after the sperm has fertilized the egg. We were all about 50 to 150 embryonic stem cells. And what can happen is that if women have undergone IVF treatment and they've maybe got a number of fertilized eggs, some of them are frozen, they could potentially donate some to science potentially. This is the kind of stage of life that we're looking at. As we go on a bit further here, so this is implantation into so the fertilized egg is moved down from the fallopian tube. It's implanting into the lining of the womb, and this is a bit by the by, but in 25% of women they get a bit of bleeding as this occurs, and that can be the first indication of pregnancy because it's not at the normal sort of time of the sort of menstrual blood. Down here, this is stage 6. So what's that? I can't quite read that. It's 13 days, I think. So this is when it begins to differentiate. So you've got the baby, placenta, and yolk sac, which looks a bit like that. So quite early on. And again, what is this at this point in time? Is that a baby? I'm sure you have your own thoughts on whether that constitutes human life at that point. So moving on down here, so this is about the time you would maybe find out you're pregnant. The period was, period is beginning to be late, and this is now visible in ultrasound. So I put baby question mark. Is that human life again there? And this is 42 days, and this is 7 weeks. So most miscarriages have occurred by this point, which I suppose, if someone suffers a miscarriage, I don't know if it's any sort of consolation that, certainly it's still a very early stage of development at this point in time. So here's just a few questions, something for you to have a think about. Hopefully we can get back in and have a discussion about this in groups in the class and as a class together. But that was just sort of related to some of the things that we've been talking about. And that's time for another break. And we'll come back and we'll do the third part of this shortly. So that I've kind of put two together here because basically this one, unit 1.5, structure of the genome, I would do it in maybe about 20 minutes in class. There's not very much in it at all, so I've just put it together with stem cells. So we'll just have a look at this. As I said, there's not much in this one. So this is kind of going. We've talked about stem cells, and we're almost kind of jumping back a wee bit to some of the DNA stuff that we did before, and introns and exons and things. So the first thing is just to define what the word genome means, and then we have coding sequences within the genome. They're your genes, but they're also non-coding sequences. So I want to have a look at what they do, or what they potentially do. Again, another nice wee video, so I'd like to go and have a look at that. So if you just zoom in on your genome. So the genome, this is the genome here of a male human, 23 pairs of chromosomes, XY, and as a definition, the genome is all the genetic information encoded in the DNA of an organism. So a human genome is in 46 chromosomes, chimpanzee 48, etc. But it's just basically everything that's contained within those chromosomes. And you remember from the topic before, during transcription, you have got introns and exons, and then the introns aren't used. And so you just end up with the exons, which are used to make the protein. And as we go through the course, people often ask, well, what's the deal with the introns then? Are they just rubbish? Why are they there? And it's a good question, and scientists are not entirely sure of the answers yet, but we can talk a little bit about what is known. So yeah, genome, you've got sequences which code for proteins, exons, and your non-coding regions, which are introns. So the non-coding regions are the majority of the eukaryotic genome, so that's 97%. So a lot of it is just junk. It's not used for anything, and they say it's sort of strange things in there, like viruses that have become part of our DNA that have mutated and now just don't do anything. But it's a whole kind of history of 300,000 years of our evolution, and a lot of it is just not required essentially to make protein. So 97% of it non-coding regions. What might happen to it? So it could be transcribed into RNA, but not undergo translation to protein. So that's your introns, and they might also have a role in regulating transcription. So somehow they help produce the RNA. They've got some role there. And if we look at this, I just wanted to just do this as a wee bit of fun to show you, that if you were to look at the letters in your DNA, what's the actual, the useful material. So I've got 6 genes in here. I mean, you might not want to do this. It might give you a bit of a headache, but it's quite good fun in the class. So there's 6 genes in there. Most of it is junk that just says blah blah blah. Can you pick out the 6 genes? So there's the first one, thing 1. Bacon bite is gene 2. His ears are nice are number 3. Pie supper is 4. Trousers over there 5, and then the last one. Did anyone get this? It's right down there. It was ha 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 ha. So again, just an illustration of what your DNA is looks like. So you can imagine when transcriptions occurring. You've got these transcription and translation. You've got a lot of it is just being ignored, and you've got the kind of important genes within that. Again, something to remember as well, you've got 2 types of RNA, transfer RNA, ribosomal RNA, so they're the non-translated forms of RNA. So they're being produced, and we know what their use is. They're involved in the translation process, and RNA make up ribosomes, but they do not actually code to make proteins, but they do have functions. And then just to finish it off, you can have a look at this film. So YouTube, search for that, 3 Sad Surprises, and I'm just about to click the arrow key, and I think this is the end. So goodbye.